All right, we may as well get started. Good day, everyone, and welcome to CanPath's last webinar of 2023. Very excited about having our two presenters today. And so I'll just introduce myself really quickly. I'm Dr. Philip Balwadala. I'm the National Scientific Director for CanPath, the Canadian Partnership for Tomorrow's Health. And thank you all for joining today's Trainee Research webinar. So today we're going to hear from two trainees who will share how they use CanPath data to explore interesting factors and questions around age, ancestry, lifestyle factors, and the environment, and their impact on health, gene and biomarker expression, and disease development as well. So we're going to have two presentations today. The first one will be by Dr. Michelle Harwood. So important to mention there that Dr. Michelle Harwood is no longer a trainee. In fact, she was a trainee in my lab for many years, not that many years, but a number of years, and recently graduated in September with a PhD from the University of Toronto. And her work is on uncovering the role of allele-specific expression and healthy aging, disease progression, and treatment outcomes. Our second presentation will be from Umaimah Zanif, a Master of Science student at the University of British Columbia under the supervision of Dr. Parveen Badi, who's our Scientific Director for the BC activity of CanPath, the BC Generations Project, and Dr. Rachel Murphy, who's also faculty at UBC. And she will share how two biomarkers linked to colorectal cancer progression connects with demographic, health history, and lifestyle factors. After each presentation, we're going to open it up for questions. So after Michelle's presentation, I'll open it up to questions, and after Umaimah's presentation, we'll open it up to questions there as well. And so please use the Q&A button at the bottom of Zoom for your questions. Chat won't be enabled. Before we start with Michelle's presentation, we're going to begin with our land acknowledgement. So we acknowledge the traditional territories of the Mississauga of the New Credit First Nation, Anishinaabe, Wendat, Huron, and Haudenosaunee Indigenous peoples on which the CanPath NCC Coordinating Center at the University of Toronto now stands. The territory was the subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Iroquois Confederacy and the Confederacy of the Ojibwe and allied nations to peaceably share and care for the resources around the Great Lakes. With CanPath teams across Canada, working from coast to coast, we also acknowledge the ancestral territories that are home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. Michelle, I'm going to now be turning this over to you. So very excited to have your presentation. And I believe, are you in control of the screen?
Yes, I will share my screen now.
Okay, fantastic. So take it away, Michelle.
Great. Thank you so much for having me here today. I'm really excited to talk to you about some of my research from my PhD. So as Philip mentioned, I was in his lab for my PhD and just recently graduated. This is at the University of Toronto and the Ontario Institute for Cancer Research. And so my PhD really surrounded the concept of allele-specific expression. And so I'm going to show you some of the work today surrounding allele-specific expression and how it may contribute to healthy aging, disease progression, and treatment outcomes. So to start a little bit broader, we know that there's a lot of diversity in these human populations, especially when it comes to various health-related measurements and disease progression. But it's quite interesting because some individuals can still have the same genotype, but still display these highly variable phenotypes. And so it's important to look at this sort of population genetics lens here to understand what's going on between that genotype and phenotype relationship. And so one interesting phenomenon, as I mentioned, I studied through my PhD, is called allele-specific expression, or ASE. And this is where at a particular position where we have two alleles, so those are heterozygote positions, we may not actually have equal expression of those two alleles. So we may have, in this case, allele 1 has more expression than allele 2. And this imbalance is known as allele-specific expression, or ASE. But why do we actually care? So let's take this example where we have allele 1 in red is highly deleterious, has some sort of disease association. In this top individual, we see that there's an overexpression of this allele, and therefore that individual may have this disease or be at high risk for this disease. Whereas in the second individual on the bottom, they have the exact same genotype. They still have this highly deleterious or disease-associated mutation. However, the ASE is actually underexpressing that damaging mutation, and we may actually see a healthy individual or at least be a lower risk of disease. So my PhD project was largely looking at why do we see this variation and these differences across individuals? Why do one individual is going to have overexpression of that harmful mutation, whereas in a different individual, we may see underexpression or no difference in expression at all? In addition, even looking genome-wide, why do some positions of our genome see this ASC, whereas others don't? Is it just kind of random who and where we see ASC, or there's actually patterns and factors that are contributing to this variation? And so some of these factors that I looked at and investigated in my PhD was looking at this sort of interpopulation variability. So investigating things such as your environment, your ancestry, and your age. And in addition to looking interpopulation wide, we can also look within an individual because we know that our tissues and our cells can also exhibit differences in variation even within an individual. So today's talk today is going to be focused on the age and cellular specificity questions, but I did want to mention that we also looked at the environment, ancestry, and some of this tissue-specific questions, and this was recently published in 2022 in Science Advances. So if you're interested in those aspects, I encourage you to check that out. But for today, we're going to be focusing on aging. So we know that age is highly variable phenotype. We can see that some individuals are going to be healthy agers. They can live long lives without developing chronic illness and be at lower risk of mortality compared to unhealthy agers. And in 2019, it was investigated to see if ASC changes with age. And they looked at a cohort of individuals at 70 years old, and then the same individuals 10 years later at 80 years old, and observed that there was about 2.6% increase in overall ASC over this 10-year period. So this was a great study to introduce the fact that ASC can actually change as we age. But there are a number of factors that are still missing from these questions. The first is that this cohort is from 70 to 80 years old, and so it'd be really interesting to see what happens if we expand that age and start looking at a younger age to observe over a longer period of these aging processes. And the second is the fact that this cohort was largely from individuals who are healthy or at least no reported disease information. And so it would be quite interesting in our healthy aging question is if this change in ASC as we age actually contributes to any of this variation we see in healthy versus unhealthy agers. And this is where the question that I was interested in came up: does ASC contribute to phenotypic variation observed during aging? And this is where CanPath comes in. So I'm using two regional cohorts within CanPath, one being Cardigan, the French-Canadian cohort, and the second being the Ontario Health Study. Cardigan, we use 832 individuals, and OHS, we have 350, and all participants are sampled from blood. Now, the unique thing of these two cohorts is that Cardigan, we actually have bulk RNA sequencing, whereas the Ontario Health Study uses single-cell RNA sequencing, which is quite unique for this type of cohort. So I'm going to go into a little bit more detail about how these samples were collected in the OHS single-cell study. And this work was largely done by Dr. Alyssa Bader, who is also a recent PhD graduate from the lab, and she used the Intermountain Risk Score, which is predictive of 5-year mortality risk. And this uses complete blood count data. So essentially a low risk score is equal to low mortality risk and essentially means a healthy blood. And so she used an extreme sampling approach of these OHS participants so that we could grab individuals based on their age, but also this risk score. So we'll have young healthy, young unhealthy, aged healthy, and aged unhealthy participants to compare. That is important that at the time of sampling, all of these individuals had no self-reported diseases, so we were focusing on the age with risk score questions. And as I mentioned, this study is quite unique because it actually used single-cell transcriptomics. So it starts to allow us to ask some of these questions at a cell type specific manner. Now, testing for ASC in a single-cell context is extremely difficult because we rely on the read coverage and the read depth information to do our statistical tests. So because of this, we first grouped all cells, regardless of cell type, into a pseudo bulk calculation so we can compare that to bulk in Cardigan. And then we grouped cells based on their involvement in innate versus adaptive immunity. We found that the patterns were similar within these two groups, and grouping them allowed us to increase its power to detect ASC. And so this is the complete schematic here, where we now see our pseudobulk in addition to separating by adaptive and innate immune cells. And that intermountain risk score calculation was done in both of these cohorts, Cardigan and OHS. So we have individuals young defined as less than 45 years old, age greater than 65, and then those risks, high and low risk score that I just explained. Now, in addition to the risk score, we can also try to use some of our disease information. And this will primarily come from Cardigan because we have information of individuals such as 49 that have cancer, 142 with hypertension, 53 with type 2 diabetes, and 120 with dyslipidemia. Now, in the OHS, I mentioned at the time of sampling, they were particularly sampled where they had no self-reported disease. However, 22 individuals using our current sampling have identified that these individuals have developed cancer since the time of our sample. So our current blood sample is actually a pre-cancer sample for these 22 individuals. Now, to get into some of the results, we first wanted to confirm the relationship that was previously shown where there's an increase in ASE with age, and we do in fact confirm this relationship seen here, and this is seen in bulk from Cardigan. But we do also see it in our pseudobulk from OHS, but when separating by cell type, it is only significant an increase in ASE with age in our adaptive immune cells. Now, when we start to separate it based on this risk score I've been mentioning, we actually see that low-risk individuals have the larger increase in ASE with age compared to our high-risk individuals. And again, this is seen in Cardigan, but we also see it in our OHS pseudobulk. And again, it is only significant in our adaptive immune cells. So the question there was, what could be driving this? Is it just that any increase in ASE is somehow beneficial for aging, or are there specific genes that we're picking up that are driving this pattern of low-risk individuals actually having larger increases in ASE with age? So to test this, we started to stratify our ASE sites into whether they're common versus rare. So common are those sites where there's a significant ASE event in more than 10% of individuals that are heterozygote at that site, whereas the rare ASE would be found in less than 10% of those individuals. And so in common ASE, we commonly saw that these individuals were involved, the genes were involved in immune processes, whereas rare was often involved in metabolic processes. And when we compared our common to rare ASE to our age and young individuals, we actually saw an increase in common ASE in our aged individuals. And so since this common ASE is involved in immune processes, we brought it back to this question: could these immune genes actually be the driving force of this pattern we're seeing of low-risk individuals having increased ASE with age? And so to investigate this further, we used an immune gene set from InnateDB. We had about 534 genes that overlapped this immune gene database to our genes that we've been testing for ASE. We tested per individual the proportion of ASE specifically in immune genes. So this is looking at the number of significant ASE sites in those immune genes divided by the total number of heterozygote sites in those immune genes per individual. And then we stratified out individuals that have a high proportion of ASE in immune genes as those being greater than the third quartile. And then we asked the question, are these individuals more likely to be low risk? And the answer was yes. So individuals with a high proportion of ASE in immune genes are more likely to be low risk. So this suggests that perhaps this increase in ASE, specifically in immune genes, may actually have some sort of beneficial mechanism to aging processes. Now we can also go from looking at that blood risk score to some of our known disease phenotypes, and reminded this is going back to our Cardigan data. So we have information of individuals from type 2 diabetes, hypertension, dyslipidemia, and cancer. When we look at our cardiometabolic traits, so first in blue, we see our normal or non-disease individuals. We still see this steady increase in ASE with age in those normal individuals. However, when we look at our disease individuals, specifically hypertension and type 2 diabetes, we now see a significant decrease in ASE with age. Now, if we look at our pre-cancer samples, and reminded this is from OHS now, we actually see larger increase in ASE with age in the precancer compared to normal. So it's an opposite effect to what we're seeing in our cardiometabolic traits. Now, this is not significant, likely due to the sample size of our precancer samples, but there are a few things that could be going on here. The first could be tissue specificity. Due to the sample size and the nature of it being a precancer sample, we have just grouped any individual that had a cancer diagnosis together. And so perhaps looking tissue specific based on the tissue of origin, but even looking at that tissue in detail rather than testing it in blood, perhaps might give us a different insight into what's going on. The second factor I think is involved is the immune system. I just showed you that immune genes may be primarily driving some of these patterns of increased ASE with age, and we know that there's a lot of overlap between cancer and immunity. So perhaps the immune genes in this ASE change may be acting different in these cancer patients compared to what we're seeing in normal. Now, we do see a similar trend in our cancer individuals from Cardigan. So these are known diagnosed cancer individuals where we see this again, increased ASE with age is larger in our cancer compared to normal. However, I do want to mention that all the individuals diagnosed with cancer in these Cardigan cohort are being treated for cancer, and so it's difficult to isolate what is an occurrence from the treatment separate from the cancer and the biology itself. However, the fact that we do see consistencies between precancer and cancer may indicate that there is something in terms of the cancer biology that's going on here, and it should be investigated further, perhaps in a cancer-specific cohort. However, our cardiometabolic traits allow us to ask some more of these questions in terms of treatment information. And this is because we do have information of these individuals taking treatment, some who are not, in addition to response to those treatments. So we used the lab-reported blood measurements, the time of sampling, which can help us tell response. So for example, in hypertension, we can know if an individual is taking treatment, and if they have a high blood pressure, they are a non-responder. Whereas if they're taking treatment and their reported blood measure is low, they are a responder to that treatment. But I do want to mention, so as this cohort is fantastic to be able to ask these types of questions, both on the disease, but also the treatment factor. However, I do want to mention that many of these individuals have overlapping diseases, overlapping treatments, different drug classes, and there's many different factors. And although we've tried to control for as many as we can, this is something that to keep in mind when looking at this type of data. But it is great to start to look at these associations with treatment and ASE, which has not been done in the past. So this is looking at the total proportion of ASE sites on the y-axis here, and we can split it based on those individuals taking no medication, one medication, two medications, or all three medications for these cardiometabolic traits. And there's a lot of information to take in here, but what I want you to focus on are these three box plots, because these are individuals that have a significant increased ASE levels. And these are individuals taking dyslipidemia medication, hypertension medication, or both hypertension and dyslipidemia medication. So this starts to tell us that perhaps there is some sort of association with treatment and ASE levels across these certain treatments. Now, when I separated those individuals based on the responder versus non-responders that I mentioned, we actually see consistent results between responders and non-responders, where both of these groups have this significant increase in ASE sites. And so this tells us that this may be an association with the treatment itself and not necessarily how that individual is responding to the treatment. However, we have identified some specific genes where there is ASE differences, specifically in non-responders compared to responders, which may act as an informative biomarker or potential predictive power to predict how individuals may respond to certain treatments. So with that, I would like to end with a quick summary. So I raised this question, does ASE contribute to phenotypic variation observed during aging? Why some individuals are going to be healthy agers compared to unhealthy agers? I showed that low-risk individuals have larger increases in ASE with age, and that this may be driven by immune genes, because increased ASE in immune genes perhaps is beneficial for healthy aging. I showed opposite effects, though, where we see decreases in ASE during aging associated with risk of cardiometabolic traits, but that medication may also have an association with increased ASE in some treatments. And so this study really opens the idea that ASE could possibly be used in biomarker and predictive power, both for the disease itself, healthy aging, but even in response to various treatments. And with that, I would like to finish off by thanking my lab. So thank you to my supervisor, Philip Awadallah, the entire Awadallah lab, who have all had a part in the work, but especially those in bold who had a direct hand in this project. Thank you again for having me here, and I'll be happy to take some questions.
Hi, Michelle. I'm trying to, I don't seem to have the ability to, okay, here we go. Now I do. All right, sorry, now I got my video on. All right, so thanks very much. A fantastic presentation and well-timed as well. All right, so first question that's come up, how do you interpret that those on all three medications had lower ASE?
So this was looking at, we did a proportion of ASE across those different groups, and then we did pairwise tests between the different medication groups and those taking no medication. And so we saw the significant increase in those three box plots that I showed. So the dyslipidemia, hypertension, and both of those.
Okay. And the same, I don't know if it's the same anonymous attendee, sorry. Is your medication data from CAG self-reported or obtained through admin linkages?
What I showed today was self-reported. So we do have information on what they're taking medication and what medication they're taking. And so we also tried to look at some of those drug classes that I mentioned, but it is all self-reported data currently.
And yeah, there has been some work that's been done on those self-reports with regard, I can say this about CanPath, with regard to CAG, about we've had collaborators look at those drug utilization data compared to admin linkages as well. And I'm just looking at if there are any other questions that are about to pop up. I'm going to ask you one question really quickly. So there was a publication that just came out that was talking about a particular locus that seems to control across different species the degree of ASE that we see in these populations. And when perturbed, when that gene is perturbed, I think what we saw in that paper was that you get allele-specific expression reaching kind of equivalent levels. So it kind of breaks down that evolved relationship. How do you think that relates to some of what you've been observing in some of your publications?
Yeah, so right now we're looking at these almost at an individual SNP level. So we've started to look at some relations to EQTLs, for example. So perhaps looking at this specific gene in our study, maybe it'll explain some of the ASE at other genes. But currently we've almost looked at it more at an individual SNP by SNP basis. Okay,
great. Okay, and if I'm just checking to see if there are any more questions. And if not, Michelle, I'm going to thank you once, and I'll probably thank you again later. That was a fantastic presentation, a very clear presentation as well. So with that said, we're going to move on to our next, a whole bunch of questions just started to pop up. Well, just one second. Okay, one last question. I think some of these were from before. So very interesting, but this is from Cindy Babinski. This info can motivate healthy living. Sorry, Michelle. This info can motivate healthy living and assisting people seeing how their meds are assisting them. So this is more of a comment. So, okay, great. And we're just going to move on. Sorry, Michelle. And so now we're going to come to Umaimah.
Hi, yes.
Hi, okay, and sorry. So Umaimah, I'm going to let you take it away. And I think you have control of the screen present.
Perfect. Hi, everyone. Thank you so much for joining Dr. Harwood and I in today's webinar, where we talk about our ongoing research in the field of healthy aging, disease progression, and prevention. I just want to thank CanPath again for the opportunity for talking about my master's thesis project, which would be investigating demographic, health history, and lifestyle factors in association with biomarkers of prognosis in colorectal tumors. Now, before I begin, I'd like to acknowledge that the land that we gather on is the unceded territory of the Coast Salish people, including the territories of the Musqueam, the Squamish, and the Tsleil-Waututh nations. So just to provide some background into colorectal cancer survival, it's not surprising that survival among colorectal cancer patients is closely linked to disease stage. You can see here in the image that the steep decline in 5-year relative survival rates when you move from localized disease with a survival rate of 91% to disease that has metastasized to distant parts of the body with a survival rate of 14%. But in addition to tumor stage, there's a number of different demographic, health history, and lifestyle factors that have been linked to prognosis. So starting with demographic factors, we know that colorectal cancer mortality is higher among men than in women. We know as well, age is also linked to survival. With older patients, treatments are often less effective, meaning that the prognosis tends to be worse in older patients. And with race, for example, we know that African Americans in the US have the highest mortality rates from the disease. There's actually 15% greater mortality rates seen in African Americans and white Americans. This has been actually attributed to socioeconomic differences and disparities in the access to medical care, but even accounting for all of those factors, there's a significant portion of the disparity that still remains, highlighting the potential contribution of the differences in lifestyle factors. In regards to health history, we know that having inflammatory intestinal conditions such as IBS, ulcerative colitis, or even Crohn's disease has been associated with poor colorectal cancer survival rates. And not surprisingly, screening for colorectal cancer, whether that be through FIT testing, colonoscopy, or sigmoidoscopy, is associated with better disease prognosis. Turning to lifestyle factors, most factors that have been linked to colorectal cancer incidence are also associated with prognoses. For example, smokers have a 40% increase in the risk of colorectal cancer mortality as compared to non-smokers. There's also a dosage-dependent relationship between the amount of cigarettes smoked and the risk of colorectal cancer mortality. And as another example, colorectal cancer patients who consume the highest level of fruits and whole grains have a 12 to 32% reduced risk of mortality because of dietary fiber. The mechanisms that underlie the impact of these various factors of prognoses remain uncertain, though various compelling mechanisms have been proposed. For example, differences in prognoses by biological sex may be attributed to differential impacts of the sex hormones on the immune system, and improved prognoses in association with physical activity could be related to decreases in the levels of insulin and the insulin-like growth factor. But another compelling yet understudied mechanism that might underlie these associations of multiple different factors with prognoses are differences in the expression of tumor markers that have been previously linked to prognoses. One such biomarker is SPARC. SPARC is a macrocellular glycoprotein that's involved in a number of different processes as shown on this figure here, including tissue remodeling, wound repair, cell migration, and angiogenesis. Previous studies of colorectal cancer have demonstrated that reduced levels of SPARC expression in colorectal cancer tumors are associated with poor disease prognoses. Another marker of prognosis is PD-L1, which is a ligand for PD-1, which is programmed F1. PD-1 is an inhibitory receptor expressed on T cells, and in colorectal carcinoma, PD-L1 overexpression is thought to shield the tumor from immune detection. So in turn, higher levels of PD-L1 expression in colorectal cancer may also indicate a more immunosuppressive environment, potentially leading to poor prognosis. Which brings me to the objective of my pilot study, which is to explore the association of demographic, health history, and lifestyle factors with SPARC and PD-L1 expression in colorectal cancer tumors. By improving our understanding of these mechanisms, may actually offer insights to inform new strategies to inform colorectal cancer prognoses. As a secondary objective, we did explore the effect of these factors in normal tissues, but for the purpose of today's webinar, I'll only be presenting all the tumor tissue findings. My study is nested within the BC Generations project, which is one of CanPath's regional cohorts. BCGP includes nearly 30,000 participants who have provided detailed demographic, health, and lifestyle information at the time of recruitment. And which brings me to one of the unique aspects of my study is that I'm able to examine associations between these factors that were assessed pre-diagnostically with markers of prognoses. rather than factors that were assessed post-diagnostically, which other studies of this type have generally been limited to. Within BCGP, I help establish a virtual tumor biorepository, which facilitates access to tumor samples that are collected as a part of a routine pathology review from incident solid cancer cases that were diagnosed among BCGP participants. Now, when I started the study, there were 115 incident colorectal cancers that had been diagnosed in BCGP. In the virtual tumor biorepository, we identified resection pathology reports for 69 of those cases. For the remaining 46 cases, we either didn't have a report or the report was from a biopsy case, which would not have had sufficient tissue to support our study. An additional 8 cases were then excluded because they were high-grade rectal tumors for which the standard of care was neoadjuvant therapy. And we know that neoadjuvant therapy could have impacted expression of the tumor markers. We then requested H&E slides for 61 cases, 7 of which were not available for various reasons from the provincial path labs, leaving us with 54 cases. Dr. Peter Watson, who is CanPath's Biosample Standing Chair Committee, reviewed the slides from these cases to identify the specific formalin-fixed, paraffin embedded, to the FFPE blocks that were needed to request to obtain tumor samples. We received blocks from all 54 cases. The tumor tissue could not be identified in the blocks for 5 additional cases, leaving us with a total of 49 cases in our study. Dr. Watson's group at the DD Research Center in Victoria created tumor tissue microarrays from these FFPE blocks. For each case, we had 2 0.6 millimeter cores that were extracted and embedded into paraffin blocks. Slides were then created from these TMA blocks for immunohistochemistry staining with the antibodies for SPARC and PD-L1. Now, I just want to give a little note here that this SPARC staining is for SPARC epithelial components. So when we look at IHCs, the slides were imaged using multispectral imaging systems, and the resultant images were analyzed with image analysis software. The software spectrally separated colors of the stain and classified tissues as epithelial versus non-epithelial or stromal. Using SPARC as an example, you can see in the tissue segmentation map that is epithelial tissue and green is non-epithelial tissue or the stromal. With SPARC, why we look at both epithelial and non-epithelial tissue staining is because even though SPARC is known as a stromal protein, much literature has shown that SPARC has functionality in the epithelial tissue of colorectal cancers as well. SPARC is known to influence the composition and function of the tumor microenvironment, and its expression in the extracellular matrix of tumors can actually affect the behavior of cancer cells, like their response to therapy. The presence of SPARC in the tumor microenvironment can actually influence how the tumor responds to chemotherapy or other treatment modalities. And for example, in a particular study, treatment of mice with SPARC conferred increased sensitivity to chemotherapy, which led to significant regression of xenograftic tumors. Moving along with the score map, the software also segmented cells according to stain intensity, which was assigned a value of 0 to 3. So you can see in the score map where blue is 0, yellow is 1, orange is 2, and brown is 3. So you can see positive yellow staining in the epithelial region. So if you overlap the tissue segmentation in the score map, you're seeing yellow in the epithelial region. Finally, the software also identified the frequency of cells with each value of stain intensity. So the phenotype map, that showed cell expressing SPARC in brown, and those cells that did not express SPARC in gray. The frequency was multiplied by the intensity to calculate an H-score, or as some like to call it, an image analysis score, separate from epithelial versus non-epithelial tissue, which can range anywhere from 0 to 300. 0 being 100% of the cells staining 0, and 300 was 100% of the cells with a staining intensity value of 3. If you recall, we had 2 cores for each of the cases, so our H-scores were averaged across the cores. For data analysis, we did do linear regression analyses to evaluate the associations of our dependent variables with non-transformed SPARC and PD-L1 H-scores in our epithelial and non-epithelial tumor tissues.
Umaimah, sorry, is it possible for you to speak more into the computer? We're having a little bit of trouble with the sound.
Oh, for sure.
Yeah.
Are you able to hear me a little bit better now?
I'm hoping that speaking to more closely to your microphone might help, but so.
Here are the distributions for some of the factors that we evaluated. Given the small number of cases, we limited ourselves to dichotomizing most of our variables. And if you can see here, we only had 3 non-white participants, which actually limited our ability to examine the impact of ethnicity on our markers. We did, however, look at variables like BMI, age of diagnosis, sex, household income, education level. Here are the rest of the variables that we evaluated. We looked at variables like colorectal cancer screening history, we looked at hours slept per night, we looked at fruit and vegetable consumption, as well as the frequency of alcohol consumption, or if a participant ever has smoked 100 cigarettes in their life, and the total physical activity minutes per week. Before we started our linear regressions, we wanted to look at the distributions of our average H scores to check for normality, and saw that our distribution was quite skewed, which made us long-transform our variables when running the linear regressions to then exponentiate the results. When running unadjusted linear regressions as Spark average H scores as the outcome, we found that individuals with incomes that were greater than $75,000 a year had 33% higher Spark H scores in the stromal components of their colorectal cancer tumors as compared to those who had less than $75,000 a year. We also found that compared to non-smokers, smokers on average had 47% less Spark H scores in their epithelial component of their colorectal cancer tumors. Now, going back to our literature, we know that lower Spark expressing tumors are known to have lower prognosis. When looking at unadjusted models with PD-L1 as the outcome, we found that compared to males, females had 2.8-fold greater PD-L1 H scores in their colorectal cancer epithelial tissues. We also saw that compared to those without any history of colorectal cancer screening, so whether that was FITs or whether that was colonoscopies or sigmoidoscopies, we found that those with a history of screening of the disease had a 2.2 and a 2-fold greater PD-L1 H scores in their epithelial and non-epithelial tumor tissues, respectively. We did not find any other significant observations that were observed, and we did evaluate the potential for confounding by adding other factors one at a time to these models, but the point estimates of interest were not meaningfully altered, so we did leave the models unadjusted. When we look at study strengths and limitations, the major study strength here was that there was a great availability of detailed pre-diagnostic data on a variety of the demographic, health history, and lifestyle factors. And that was ascertained from the BCGP Health and Lifestyle Questionnaire that was done at baseline. We know that pre-diagnostic data actually allows us and enables studies of early intervention, risk management, and the development of targeted strategies like prognoses to improve health outcomes for individuals and the population. Key limitations in my study included the small sample size of the study, which was driven by the substantial number of cases that we needed to exclude, which was about 57% of our cases. It's possible that reasons for exclusion are related to prognoses, which could and would impact the validity of our results. And as well, tumor stage is the major predictor of prognosis. We did not have full access to stage information. We were only limited to whatever the CAIS had within the histopathology reports. So if the histopathology report specifically had its tumor stage, we could confer that that was the tumor stage. But if a path report just explained the infiltration of the tumor into different tissues and lymph node involvement, we know from talking to pathologists that we cannot ascertain tumor stage until we have other reports knowing if there was distant metastases in the lymph nodes. Which brings me to the conclusion of our study, which is that larger scale studies with prognostic data or survivorship data are needed to confirm our findings, but our results suggest that the differences in expression of Spark and PD-L1 may contribute to the previously observed impacts of subdemographic, health history, and lifestyle factors on colorectal cancer prognosis. And with that, I just want to thank everybody for showing up today, as well as I want to give a big thank you to my supervisors, Dr. Kevin Betty, Dr. Rachel Murphy. I want to thank the rest of my master's supervisory committee, as well as our team over in Victoria, who were able to help us create the TNEs, to do the IHC staining for us. I also want to thank our fellow collaborators on our biorepository paper, as well as our BCGP team and our participants. Thank you so much.
Thanks, Umaimah. I'm just waiting for permission to turn on my video. Okay, now it's allowing me. Okay, very good. Thank you very much. Thanks, Umaimah. A very clear presentation. Opening up this for questions presently, we have a comment. So thanks for this very informative discussion from Sue Street. While we're waiting for any questions that might be coming in, I'm going to ask a couple if that's okay. So my first question is why, you mentioned at the very end of your project that you had to exclude, I think it was around 57% of cases from the study. Was that in part because of the second point in terms of not having enough pathology information, or was that something distinct?
So there were a variety of reasons why we excluded. At each stage. There was different things that we ran into. So when we first looked into the virtual tumor biorepository, we found that within the CAIS, there was a number of cases that we could not find reports for, and that was due to a malfunction within the CAIS. We also had to exclude further cases that just had biopsy reports. We know that if cases just had biopsy reports, that did indicate distant metastases. So that would mean that there was no surgery done because that case was a metastatic case. We also found that when we were requesting cases from different path labs, we were finding, for example, with Northern Health in BC, they had staff shortages. So that means 7 of those cases were ineligible for us to obtain from there. We also did find when we did tumor tissue for the TMA, some cases, even though they had FFPE blocks, they just didn't have sufficient tumor enough for those cores. So at different stages, there were just different things that we ran into.
Okay, great. I have a second question. So I'm just thinking about, there's a lot of excitement these days about how quickly we can get something into a clinician's hands so that they can be more aggressive with regard to treatment following a diagnosis and something like that. So very recently there was a particular paper that's got a lot of excitement that people are able to now, even in the surgery, almost do something like a biomarker screening. In that case, it was using looking at panels of particular genes. In this kind of context too, are we thinking about using Spark and PD-L1 from like even in the hands of a pathologist then? Are we thinking about it in that kind of context too, like in terms of a prognostic or in terms of like a diagnostic in terms of how that clinician is going to be using that information?
I think to answer that question fully, I think we definitely should start with prognostic just because larger scale studies would allow us to be able to ascertain what we found here with the 49 cases, to be able to then circulate through clinicians' clinics and then do nested case control studies and then be able to get a direct answer.
Okay, super. Okay. We have a question from Cindy Babinski. Any ideas on how to access more clinical data?
Yeah, so that's a really great question. Within the CAIS, currently we are limited to whatever is in there. So we have histopath reports. Now, if we wanted to get more surgical reports and even more doctor commentaries or addendums, we would need to have connection to Care Connect, which is what one of the pathologists on my supervisory team they advised me to do, but with that you do need more ethics and you do need more. You need to go back and be able to ask consent to be able to link to Care Connect. There are differences to connect to CAIS and Care Connect.
Okay, I guess there's also potential opportunity to go to outside of the province.
Yes.
That's not an easy. That's the general Canadian problem. But the CanPath is trying to do some more. Is actually, we hope to be announcing some exciting action in that space next year. Okay. Another question just popped up. Mahmoud Tazari, are those data public and open source?
I don't know which data you're talking about.
They might be referring to any of the pathology slides or some of the images that you were showing, as well as maybe some of those outcomes that you were talking about just a moment ago.
So currently, pathology reports are not open to the general public because that has patient identifiers. So that would have to be through direct consent, and through BCGP we have that. With the TMA data and the IHC staining, that's actually a resource that we created within BCGP. So that would be available in after my thesis is published as a resource for other collaborators to be able to go into the TMAs and stain for different things.
Okay. Actually, I was just thinking about this in Ontario. There's something called the Ontario Tumor Bank. So either you or Mahmoud, for example, might be interested, and they actually, we've been utilizing that resource because they have slides. And I don't know if they're stained with the things that you'd be interested in, but it's also worth checking out because we've been looking at pulling some of those things in there. I think there are 20,000 or so images in there, coupled with biobanked material as well. So I think it's an interesting resource in that context as well.
Definitely, yeah.
Okay, and in that with that, yes, I'd like to even just kind of quickly highlight that hopefully next year we'll be able to, won't necessarily have imaging data, but we'll be able to report that we're looking at supporting a Canadian cancer study that's going to provide access to the general research community with regard to linkages to administrative outcomes. And with that note, thanks. I'm going to thank you again, Umaimah, and I'm going to thank you, Michelle, as well. Hopefully we can get your screen back up or your video back up so we can see you and thank you in person like that visually. Great presentations from the both of you. This might have been one of our records in terms of attendees for a trainee, quote unquote, webinar. I should highlight also that Michelle isn't a trainee. Again, she's now at Genentech. At the Canadian office as well. So great work from both of you. And the last thing I'm just going to highlight here is our next trainee webinar is going to be in the new year on January 22nd. Stay tuned, and you'll be hearing from CanPath Communications, Megan, more about when that will be scheduled, specifically specifics about that scheduling as well. Thank you, everyone, and thanks to all of our attendees as well. And I hope everyone has a great holiday, and we'll see you all next year. Thank you very much, everyone.